I'm Ted Malinski from Ohio University, and title of my presentation was about the role of L-Arginine in the preconditioning of ischemic heart. We use nanomedical approach in the studies, and this is based on this nanomedical approach is based on the development of nanosystems like nanosensors, nanorobots, nano delivery system, which we use to monitor concentration of diffusible molecules in this particular case in ischemic heart. Primarily, we study the release of nitric oxide, which is considered a cytoprotective molecule, and we monitor simultaneously with the devices which are smaller than about 1,000 times smaller than the single endothelial cells, and about 700 times smaller than the single hair. Means that we can penetrate very small volume and measure very few molecules when they are present in this very small volume of the ischemic heart. And it appears that in ischemic heart we have on the very beginning we have a very high release of nitric oxide, which is used for the protection of the system and also restoration of the blood flow, which is impossible to do because of the clot. And this nitric oxide release is followed by the release of the oxidative stress molecule, and these are superoxide and peroxynitrite, which is a very, very oxidative molecule. And in the same time, the system tries to adjust to the very unusual condition of the low oxygen, and will start to improve the L-Arginine transport. And arginine is a substrate for the synthesis of nitric oxide, and is in relatively high concentration, but we are in the cardiac cell. But we are not looking for absolute value of the concentration, which is more than sufficient to produce nitric oxide. We are looking for enhanced delivery of nitric oxide, and L-Arginine will be involved, higher concentration of arginine will be involved in this particular process. Means that we introduce some special scale or coefficients which show us how much damage can be imposed on the cardiac cells, and it is based on the ratio of nitric oxide to the sum of the concentration of superoxide and peroxynitrite. And the normally functional heart, this ratio is about 5 or 6. In under ischemic condition can be as low as a fraction of 1. Okay, and it is a clear indicator and correlate very well. This factor correlate very well with the amount of damage. What preconditioning is doing really is under this very stressful condition of the ischemia, is preparing more of the defense molecules, and the L-Arginine is one of them, and of these molecules, and increase in L-Arginine decrease oxidative stress, improve the ratio of nitric oxide to superoxide peroxynitrite, and limit or decrease the damage to the heart by about 50 to 60%. And it is very important process, which is natural if occurs in the case where we have some small ischemia before we get a major ischemic event. And however, this process can be mimicked by the supply of L-Arginine before an ischemic event, and it does exactly the same and improve and release and decrease nitrooxidative stress and decrease the damage to the heart. And it is our major funding in this particular research.